And the next lecture is by Siddhant Sharma and Jim Cleaves from LC in Japan. And the title of the talk is a computational simulation of reaction networks involved in life essential reactions.
Just a little bit introduction about myself. I'm a bachelor student in India, and my concentrations are in biochemistry and chemistry. And I did this work as a part of our summer research project with Dr. Jim Cleaves and other people at LC and BMSIS through their programs last year, and it continued through a year-long project. And just for acknowledgements, here are all the wonderful people that were involved with me, a lot of undergraduates, a lot of master's students. And I would really love to acknowledge SETI Forward Award that funded a part of my research. Now, why Origins of Life? Origins of Life, as you know, the Israel Society has been sponsoring Origins of Life related concepts, and it interested all of us who are present here, and it interests me too. So we thought to develop an in silico model where you can input a bunch of molecules. It will throw out a lot of results, such as autocatalytic networks. And I'm really thankful to Dr. Semenov for describing autocatalysis in an experimental setting. And I'll be talking about it more theoretically. Most of the work is computational, most of the work is theoretical while matching experimental systems. Moving on, just to give you an account of our study workflow, in the start of our project, we studied alkaline degradation of glucose in aqueous environments and Formos reaction. As you all know, Formos reaction is a well-known example of autocatalysis, and it served us well. So what we did is we used, in a center of our theory, in the center of our experiments, we used graph molecular language, GML, and graph theory-based systems such as MOD, which is developed by our collaborator in Southern Denmark University. What MOD does is it defines the rules. Rules, as you know in life, as you know in chemistry, is how your molecule will behave. So we define our molecule in SMILES, apply those rules, and build a network of molecules using MOD. Then we remove bad structures, the structures that we don't want in a huge network, since it's all theoretical. In an experimental network, you can control pH, temperatures, and pressures, and remove some of the bad structures. And in a theoretical network, it can blow up quickly. It can take a lot of computational power. So what we did is we removed the bad structures, and then we did some descriptive analysis of our molecules. Moving on, the bulk of the topic is autocatalysis, and we model autocatalytic cycles, whatever alkaline degradation of glucose and Formos were producing theoretically. Now, just a little bit introduction about MOD. As you know, it's a graph theory-based system, and as you can see on my screen, it's a basic rule of how a chemical transformation occurs. You have L, K, and R, which is left, which is reactants, products, and K is the constant in German, which is the transition state. You just define the rules, which protons you want to subtract, which bond you want to add, and this is how it processes stuff in graph theory. Due to short interest of time, I don't think I'll have a lot of time to delve into more theoretical methods in graph theory, how we developed these rules and how we applied them. Now, the Tottenham problem. Yes, tautomers, when we were starting with our rules, a lot of the generational diversity it was generating were tautomers. And we didn't want that. We didn't want our models to generate tautomers. So there were two methods that we could have used. One was as and when the tautomers were increasing, because it can blow up the network, and we don't have computing resources such as a supercomputing resource with LC or something else. We didn't have that much of computational power. Either you just clean the tautomers as it comes, or you can tweak the rules. You can modify your rules such as basic organic chemistry rule, Michael addition, Wengel, your tautomerism rules, ketonol, etc. So we cleaned the tautomers. As and when the tautomers were coming, we chose the most canonicalized form based on the stability using Ambit and RDKit, very common computational chemistry packages. But then we found out it's a really tedious exercise. As and when your network is exploding, it's a very tedious exercise. So we went back to the drawing board and modified our reaction rules in a way that it doesn't produce any tautomers except for one main structure, which is mostly stable, highly stable, which we can manage. Now, what we did, is this a reliable technique? And is this reliable in the case of matching with the organic meteorites, like matching with the organic meteorite diversity that is developed by, say, people such as Dr. Copeland in carbonaceous meteorites, or Murchison meteorites, or even Formos chemistry, which can be a definitive way to proceed with evolution of origins of life. Yes, the reaction rules were very constrained, and they were developed based on chemical intuition. Yes, the methods were semi-empirical, and they were not based on quantum chemistry methods, which definitely require a use of supercomputer. So that's a, you can say, trade-off that we made. Post-generation filtering was done, as I was mentioning about batch structures in organic chemistry, carbons with double bonds, two double bonds, and which have 3 or 4 members, which are really unstable. As if you compare it with carbohydrate chemistry of glucose and formose, they were neglected and they were left out so as to maintain a generation diversity to further calculate the autocatalytic cycles that I will be talking about. I personally believe that you should compare your theoretical models with experimental systems, and experimental systems definitely do have more messy chemistry, but they provide a really ground truthing. So we compared our glucose results to a very good paper by Yang and Montgomery, and we were able to match 96% of our molecules just by using the structures in an SDF file. Comparing it with formose structure, which is given in experimental system by Decker and Omran and other people, we were able to match 100% of our formose systems, and that was a win for us. 96% of the glucose molecules, we are still evolving our rules, we are still matching our process so as to make it a more robust system, so as to match 100% of the molecules. Comparison with the high resolution mass spectra data, the mass spectra data was calculated of alkaline degradation of glucose and formose, and even with organic meteorites with our collaborators, Dr. Human Chan at National High Magnetic Field Laboratory in Florida. So we try to match the systems that we have in our theoretical way to the experimental ones that were coming from Florida, and we were able to match a lot of them, basis on the depending on the basic of MS cutoffs that we fitted in our theoretical models. Now, yes, I want to discuss the network statistics, as I kept mentioning about the network keeps blowing up. How does the network blows up? As you can see, each iteration is called a generation, such as if you have 15 molecules in an initial starter set could be glucose and water. If you put it into your graph grammar based model, the initial generation would be 16, and then the reactions each generation will start to blow up. As you can see at the evolution of time, in the 5th generation, you have 40,000 molecules, which is a lot if you don't have a supercomputer or a supercomputing facilities, and it took us 2 days to model that. For formose, we were able to run up to 6th generation, one more than glucose, and the result was 20,000 species. And in that species, we are finding the autocatalytic cycles as the case with glucose. Now, the fantastic autocatalysis loop and
you have two more minutes to go.
Okay? Yeah, I'm finishing up. This is my last slide. And how did we model autocatalytic loops and feedback routes? We use a graph-based database such as Neo4j, which was able to find a pattern matching system. More experimental systems were discussed by Dr. Sergey, and I don't think a really huge need is to tell them about experimenting. Use pattern matching in the Neo4j system so as to maintain how the autocatalytic cycle is evolving. And that's how we were able to figure out the crucial pathway of autocatalytic cycles, especially in formose networks. Now, just the improvements. How could we improve? What can we improve? We took out the thermodynamic parameters. We used group contribution methods as compared to DFT, which are a lot less semi-empirical. How do different reaction networks converge? How do different prebiotic pH, temperatures, pressures? We never look into account that, and we would love to take into account in the future. Feasibility of autocatalytic cycles, yes, for how many of them are good candidates actually. And solving the stereochemical black box, I never talked about how we employed stereochemistry, because modeling stereochemistry theoretically is very a tedious process. So that brings me to the end of my talk. If someone is attending Abrat, Pond or Goldschmidt, definitely love to visit me. And if you have any questions, here's my Twitter.
Thank you very much, Siddhant.